So now I will not wait more. So I welcome all the participants and today's speaker, Dr. Ankit Rai ji. As you all know, this is Science Dialogue Series, free webinar series. Purpose is to increase overall knowledge pool of the society. So I'm grateful to today's speaker. He is assistant professor at Gujarat Biotechnology University, and he is also Ramalingaswami fellow. ये एक ऐसा फेलोशिप है जहां 3-4 साल 5 साल पोस्ट डॉक करने के बाद ये रिटर्निंग के लिए गवर्नमेंट ऑफ इंडिया एक स्कीम लाई है जिसमें 1 लाख के आसपास फेलोशिप देती है। अगर ये परमानेंट फैकल्टी नहीं भी होते हैं तो भी 1 लाख के आसपास इनको फेलोशिप मिलता है टू वर्क ऑन रिसर्च प्रोजेक्ट। ही हैज ऑल्सो पब्लिश्ड सेवरल पेपर, रिसर्च पेपर। वन इज इन पेनास ऑल्सो, बहुत रिप्यूटेड जर्नल है उसमें। And he's working in medical biotechnology aspect. So today he will take on that cancer, one aspect of cancer. So I welcome Ankit Rai ji in this science dialogue series. Now platform is open for your talk.
Okay, sir. Thank you so much. First
Yes.
of all, thank you so much. It's my pleasure to be part of this webinar series, and thank you so much for inviting me for this panel.
आवाज
And
थोड़ा सा वॉइस अगर और पास में हो जाए थोड़ा सा।
अभी ठीक है, sir?
Yes, it's good.
So good evening, everyone. So myself, Dr. Ankit Rai. Briefly, I will give you my, like sir already introduced, my MSc is from Pune University, then my PhD is from IIT Bombay with Professor Bilal Panda. So that lab is known for cell cycle, cell division, and cancer drugs. And after doing my PhD, I went to Netherlands to Utrecht University. There I was doing my postdoctoral research for almost 7 years. There also I was involved in cancer drugs which are targeting cytoskeleton and try to understand their mechanism of action and everything, which I will talk about shortly. So I will be very happy to answer any questions in between, or after that, just stop me in between so that if you have any doubt. I hope my screen is visible now.
Yes, full slide. So
And
yes, now it's visible.
okay. So currently, actually, I'm working as an assistant professor at medical biotechnology at Gujarat Biotechnology University. So briefly, I will also introduce my university. It is one of the very unique university designed by government of Gujarat, which is basically
But
focused on.
many speakers from Gujarat Biotechnology University has
Yeah,
delivered.
I
Yes,
know. So that is
yes.
perfectly fine. Yeah, I know. So uh, it is basically focused on totally on medical biotechnology, situated in a very nice location at Gift City, Gandhinagar. And today's presentation, I will talk about the cytoskeleton dynamics in cancer chemotherapy. Okay. So we all know this the beautiful picture of a cell, like we all introduce itself as a basic unit of life. And when we say a student to draw a cell, they beautifully draw these pictures. You know, they put somewhere the nucleus, then mitochondria, endoplasmic reticulum, ribosome, everything inside the cell, and with their positioning and everything. But I don't know how many of you ever thought, like, suppose this is the cell, and you start putting things inside the cell, like, have you ever put your attention that how these organelle inside the cell get positioned? Like, what is making the nucleus to be somewhere here, or ribosome somewhere here, or endoplasmic reticulum somewhere here? So, like in human body, we have bones and muscles which provide structure and strength to our human body. Okay, so similarly, in cells, we have cytoskeleton. Okay, all cells, including bacteria, have a cytoskeleton in one form or another, which provide mechanical and physical strength for cell survival. And I don't know if you know, in bacterial cell, the the protein which is maintaining this architecture and provides cell division is called as FTSG, okay, which is a filamentous temperature sensitive mutant Z, which is one of the very nice focus of today's bacterial cell division field and things like that. However, in my focus will be on the mammalian cell. In a human or in mammalian cell, cytoskeleton is a highly dynamic network of filamentous protein that link all regions of component of the cell. Okay, and based on their size and shape, the cytoskeleton is divided into three well-defined filamentous structure. And the smallest one is actin, which is around 7 nanometer in diameter, and its monomer form is known as actin. G actin and F actin, I think most of you have already read in your masters, if I'm not wrong. Like the globular actin is the monomer form, and when it's make these filamentous structure, it is called as F actin, filamentous structure. Then the largest subunit is largest cytoskeleton is microtubule, which is around 24 to 25 nanometer diameter, and they are made up of alpha and beta tubulin heterodimers. Okay, one thing I would like to focus here that these monomer subunit, which is a heterodimer, is always present in the form of alpha and beta tubulin. These subunits, these are dimer called as one monomer or heterodimer. Alpha alpha and beta beta interactions are very are impossible because they are not favorable. However, in between, like actin and intermediate, there are several intermediate filaments which size range from 8 to 12 nanometer, and they have different. There are class of intermediate filaments. For example, vimentin, nuclear lamines. These are all comes under the intermediate filament section. One thing also I will emphasize here, like a lot of research has been done towards actin and microtubule. However, a very less things are known about the intermediate filament. So in today's field, people are focusing more and more towards intermediate filaments because in the beginning it was thought they are not doing anything else, but they are just playing as a supporting role for actin and microtubule. But with research and with the progress of microscope and fluorescence spectroscopy, live cell imaging, it is very evident that intermediate filaments are also playing very crucial role in the cell architecture, cell function. Okay. However, my research interest lies towards microtubule, which is, as I already said, made up of alpha and beta tubulin heterodimer. Both subunits are 50 kilodalton protein, and they are synthesized on separate gene. They have different isoforms also in cell. So that is why one of the very major challenge in the field of cytoskeleton, if you want to mutate or delete this protein, it become very it's kind of impossible because if you delete one isoform, another isoform comes into the picture and take the function of that isoform. So there are six to seven isoforms are known for alpha tubulin, and there are six isoforms are known for beta tubulin. Both subunits are synthesized on separate genes, and when they are synthesized, they come together to make an alpha and beta tubulin heterodimers. Both are 50 kilodalton. One of the important aspect of this heterodimer is that they have a GTP binding site. Alpha subunit has a GTP binding site, beta subunit has a GTP binding site. However, when the heterodimers come together and make a heterodimer, the GTP which is present at alpha subunit get embedded into beta subunit. Okay, and because of that, it become inaccessible for any other interaction. So it is called as unaccessible GTP, whereas GTP which is present at the beta subunit is accessible for the interaction and other functions. So what happens that in the presence of GTP, these heterodimers associate on top of each other by taking GTP as an energy source. So in case of actin, the energy source is ATP, whereas in case of microtubule, the energy source is GTP. So in the presence of GTP, these heterodimers associate on top of each other at head to tail fashion to form a one protofilament, and these 13 protofilaments associate each other to form a microtubule. you need to also remember that these microtubules are structurally and functionally polarized structure. For example, when they associate head to tail fashion, the one subunit, one end is always having alpha subunit, another end is having beta subunit. So the end which is having beta subunit is called as plus end, and another unit is called as minus end. The reason for this designation is that the subunit interaction from beta subunit is much easier. So polymerization from this end is become much more easier than this end. So that is why this end is designated as minus end, slow polymerizing end, whereas another end is fast polymerizing end. That's why it is called as plus end. Another thing you need to pay attention that in cells, we know that we have MTOC, which is called as microtubule organizing center, which is made up of gamma tubulin and associated proteins, which are called as GPC or gamma TERT complexes. In cells, it is present near nucleus, which is called as centrioles. Okay, so centriole behave as a MTOC where these minus end is capped and it is not polymerizing. So in cells, most of the time microtubules are only polymerizing from the plus end, whereas minus end is not polymerizing. However, with recent research, it has been proved that not only centriole but Golgi, ER, and other these some other centers of MTOCs are also present in the cell from where microtubule polymerization can take place. And in vitro, if you will see that microtubule polymerization is a nucleation driven process. So if it is a nucleation driven process, so it has a lag phase. Once this nucleus is formed on which microtubules can polymerize, then microtubule polymerization become very exponential and it become log phase. And after certain time of point, it become saturated. So both ends are polymerizing and depolymerizing. So this is some of the basic biochemistry of microtubule polymerization, that it has two ends, plus and minus end. And also one of the things which you should remember is the critical concentration of the microtubule. What does it mean? The critical concentration is the threshold concentration above which the polymerization will happen. Since plus end polymerize faster than the minus end, so the critical concentration is less for the plus end, whereas the critical concentration for minus end is higher. And if you will see the in the EM structure, the diameter of this periphery of this microtubule is around 25 nanometer. As I already discussed that microtubules are structurally and functionally polarized structure, and several motor proteins as well as MAPs has specificity for these polarized structure. For example, we have kinesin and dynein, which use these microtubule as a motor protein. These motor protein use microtubule as a track for transporting various cargo from one compartment of the cell to another compartment. Then we have also MAPs, microtubule associated protein. For example, some of the MAPs specifically recognizes minus end of the microtubule. For example, CAMSEP is one, this calmalodane driven associated proteins. They specifically binds to the minus end of the microtubule, whereas, for example, EBs or CLASP or CLIP, they specifically binds to the growing end of the positive end of the microtubule. So here one of the live cell imaging which is showing, you can see that the EBs, which is GFP tag, and these microtubules are red fluorescence. You can see that these plus end binding proteins are specifically binding to the growing end of the microtubule and tracking the end. So because of these properties, microtubules are found to be highly dynamic polymer. So microtubule has two properties. For example, actin, you must have read in your masters, if I am not wrong, that it display this treadmilling when the polymerization of one end become the depolymerization from the other end, so the net length of the polymer remains same. So in case of actin, treadmilling is very peculiar, but in case of microtubule, it exhibit dynamic instability. For example, you will see that microtubules are continuously growing and shortening, and it is a catastrophic process, random process. microtubule can grow at any time and become shortening any time. So this polymerization and depolymerization property of microtubule is called as dynamic instability of microtubule. And because of this property, microtubule performs various cellular function inside the cell. It maintains this integrity structure of the microtubule. Several motor protein used in this as a track. Microtubules are also involved in cell migration. For example, during cancer, when the metastasis happen, microtubule and actin crosstalk become very crucial at that time. However, the most important function of microtubule comes during mitosis. During mitosis, these microtubule forms spindle. The bipolar spindle and chromosomes are attached at the metaphasic plate. And once proper chromosomal alignment at the metaphasic plate has happened, bipolar structures are achieved. These microtubule helps in the chromosomal segregation, which ultimately leads to the cell division. Okay, so microtubule is one of the most crucial important participant in the cell division. And because of that, since microtubules are involved in so many cellular functions, and we know that cancer cells are always dividing, so microtubule become one of the most important target during these processes. So if you target microtubule, cells will be blocked in mitosis, and cancer cell will not be able to divide, and that lead to the cell death activation. So that is why it has been found that microtubules are one of the most successful drug target for cancer chemotherapy. And these drugs are commonly called as antimicrotubule or tubulin binding agents, and they are widely used for treatment of almost all kind of cancer. If you will see the treatment regime for any cancer treatment, you will see that it is dominated by the microtubule targeting drugs, specifically Taxol and vinblastine binding drugs. And based on their binding, it has been found that tubulin has different binding sites, preferred binding site at tubulin structure. Three structures are very well known. One is colchicine, another is vinblastine, and next is this taxane binding site. Okay, so the taxane binding site is at the beta subunit, but it is located inside the lumen. So if it is microtubule and it is the lumen of the microtubule, so the taxane binding site is inside the lumen, whereas vinblastine binding site is at the beta subunit, but at the top of beta subunit. Whereas colchicine binding site is in between alpha and beta subunit at the interface of that. However, there are other drug binding sites are also being found. So most of the drug which has been found very successful in cancer chemotherapy, they mostly bind to vinca, colchicine or taxane sites. However, there are some unique sites are also present where these drugs can go and bind. For example, metansin binding site or lolamide or pleuroxide binding sites are also there. Okay. And based on their mechanism of action, these drugs were classified into two groups. One is called as microtubule stabilizing drug, whereas another group is called as microtubule destabilizing drug. Microtubule stabilizing drug, such as Taxol or epithelone, they stabilize microtubule, they enhance microtubule polymerization, or they does not allow microtubules to be dissociating, undergoing depolymerization. Whereas microtubule destabilizing agents such as vinblastine, colchicine, they depolymerizes microtubule, they inhibit microtubule assembly, and they do not allow polymers to be polymerizing. Okay, and they have different way of doing it. For example, vinblastine binds to the end of the growing polymers and does not allow another heterodimers to come and bind. It is called as end poisoning of the polymer. Whereas colchicine, what colchicine does, it binds to one of the heterodimer, and when this heterodimer is going and binding to the polymer, next monomer is not able to come and bind it. So this is monomer sequestering kind of thing. So in the same way, Taxol has, like, some of them they just bind at the end and they does not allow. For example, maps, if I will talk about tau. So if you have these two, so two protofilament, these maps can act as a glue. So they don't allow these protofilaments to fall apart, and that's why they stabilize. Or some of them can reduce the critical concentration for the polymerization. For example, Taxol and all, they binds at the tip and does not allow and allow more and more polymers to bind, which I will talk about. Okay, so these are some of the drugs which I have listed here and been shown like they are in the treatment for cancer or different clinical trials and everything. However, has been also very evident that the resistance are also getting developed for these microtubule targeting agents because of various reasons, because of the mutation in tubulin gene, or because they are now the target of efflux pump, or there are a lot of side effects, like, so for example, since uh, microtubules are present in normal cells also, so the side effect is the toxicity is one of the reasons. Because of that, uh, development of drug new drugs and everything, and understand their resistance development become very important part of the cancer research. One of my research where I have done, I have developed the colchicine resistant cell line. The methodology which I have used was like I started slowly growing the cells in a very small concentration of drug, and with time I have found that suddenly there are a lot of cells, they are now resistant to colchicine. They can now even grow in a very high concentration of colchicine. And then I started understanding their mechanism of action, how these drug and these resistant cells have adopted or grown the resistance towards colchicine. And what I have found, two things I have found. First is that there is an increase in the beta 3 tubulin isotype. So now, you this one of the very important part of tubulin research is that different drugs have different affinity for different isotype. So it is possible that one drug which is binding to beta 1 tubulin, it might possible that it will not bind to beta 2 or beta 3 tubulin. And that is why it is very important when you are designing the chemotherapy with respect to microtubule targeting drugs, because different cell type, for example, skin cells may have different tubulin isotypes or composition of tubulin isotypes compared to liver cancer or lung cancer or something. So while designing, you need to know the cellular composition. Another thing, like why personalized therapy is very important, because it might possible if two-person is having same cancer, but their isotype of tubulin can have different compositions. And because of that, when you are designing the drug chemotherapy, you need to pay attention with person to person. So that's why when you are designing chemotherapy, it might be very successful for one person, but it is creating toxicity or side effect to another person. So one of these finding was that overexpression of beta 3 tubulin isotype is not allowing the colchicine to bind now to the microtubules, and because of that, cells are developing resistance, which was also evident when I did the live cell imaging for the microtubule dynamics. It has been already reported that beta 3 tubulin isotypes are less dynamic as compared to uh, uh, other isotypes, which was also evident because there is more beta 3 isotypes. So there is microtubules are less dynamic compared to control. Also, another research, so another two of my research during my PhD was to do target based drug discovery by screening the libraries of synthetic compounds. What we do, or it is in most of the drug discovery research people do, is that they have a library based on their structure function analysis. People synthesize these libraries thinking that these molecules can have chemotherapeutic potential. So in my case, I have selected rhodamine and benzimidazole because these moieties have been shown very promising in chemotherapy, especially in terms of cancer chemotherapy. And then cell-based screening was done. So I have tasted at one particular concentration, I have tasted these drugs, whether they are able to inhibit cancer cell proliferation or not. So those cells which inhibited cancer cell proliferation, like they are showing anti-cancer activity, the next step I have checked whether they can block the cells in mitosis or not. What I have found, so the reason for doing this was that if cells are targeting microtubule, they will block the cell division, cells will be blocked in mitosis, and ultimately cell death activation will happen. So one of the peculiar part of this, that if cells are blocked, it might possible they are targeting cell division machinery, maybe microtubule or mitotic kinases. So next step, those compounds which block the cell cycle progression at mitosis, I have selected those compounds and tested in vitro on the microtubule polymerization. And by that, those I found discovered two compounds, VCFNP and CXI-benzo-84, which showed very uh, high anti-microtubule activity, and they were found to inhibit microtubule. For example, you can see in control, microtubules are highly dynamic, but when you treat with these drugs, microtubule become very static. Okay, so the and also one of the important or what I can say is the finding was that the screening was done on the resistant cell line. So these drugs were found to be even active in case of those tumors where resistance was reported for the known inhibitors. So this work was also highly appreciated by Nature Asia at that time, and the patent was also filed. And with that, I have also got best thesis award from IIT Bombay, and then I moved to Utrecht University, Netherlands, where I started doing my postdoctoral research, where I was doing the visualization of microtubule drug interaction in real time at the level of single filament. So, why it is important to do microtubule drug interaction in real time? The reason is very simple, that whatever experiment till now, most of the research has been done on the cytoskeleton field, or even I can correlate to other fields when we are doing at the bulk assays. The concentration of drug which we used in test tube when we are trying to understand its mechanism of action and things like that, we use very high concentration of drug. The reason is because the sensitivity of the machine or the sensitivity of the assay is not that much, so that we can see the effect. So to see the effect, we use very high concentration of drug. So I will take one example, for example, Taxol or Wimblastin. Okay, so their IC50, where half of the cells are died, is in nanomolar range. One is like 5 to 10 nanomolar range, their IC50 comes. However, when we test in test tube to see the interaction of tubulin with Taxol or something, we use 1 micromolar. So nanomolar to micromolar. This is a very different statistic where we are doing. At micromolar concentration, cells will even not survive. So we don't know the exact interaction mechanism by which these drugs are interacting with its target. So it is very important how one molecule of microtubule or one molecule of tubulin is interacting with drug to actually know how it is happening, so that we can see and understand the peculiarity of the chemotherapy. So to understand that, we have developed one assay which is called as in vitro reconstitution assay for microtubule dynamics. What happens that in this assay, at the cover slip, at the slides, we make microtubule polymerization. So this is one of the very complex assay. If somebody wants to understand this assay, I can, we can sit somewhere and we can explain it. But for the sake that as I already discussed, that microtubule is a nucleation driven process. We provide nucleus and we provide other accessories for the polymerization on which microtubule can polymerize, and then we perform turf microscopy. So the turf microscopy is total internal reflection microscopy. So what happens in the, it is based on the principle of turf. When a light hit at a particular turf angle or a critical angle, the total lights will be passed in the same thing. However, in this, it hit the very small peripheral sample which get illuminated, and because of that you don't get that much of background, which is, for example, if you are doing confocal or something, you hit the sample with direct light, so all the sample get illuminated and background become very noisy. Okay, so this is the principle of total internal reflection. For sake of thing, you should know. And then we perform tough microscope with all the thing you can see. So this is the cell. Okay, you can see in cell microtubules are polymerizing, and this is in the test tube. The same thing you can see microtubules are polymerizing and depolymerizing. Okay, so here what I am using the fluorescently labeled tubulin, and the plus and tracking proteins are tagged with GFP. Okay, and in the same way it is happening here, like in the absence of fluorescent tubulin, you will just see the fluorescence of GFP, the green part of the growing end of the microtubule. So because of that, so these live events of microtubules can be drawn in the form of chromograph using images software. And these, you can see this is the nucleus, and it is made up of GMP-CPP, which is a non-hydrolyzable analog of microtubule. And on top of that, microtubules are polymerizing. You can see that microtubule is polymerizing, depolymerizing, going back to the seed, polymerizing and depolymerizing.
पास में मोबाइल है क्या? कहीं फोन आ रहा है क्या आपका?
नहीं,
कुछ आवाज टाइप का हो रहा है.
अभी
वो जैसे होता है ना, जैसे स्पार्क टाइप का आवाज होता है. हाँ, अभी नहीं हो रहा है. प्लीज
अभी ठीक
कंटिन्यू.
है.
जी जी
ओके, आस हो सकता है आसपास में किसी का होगा, उसकी वजह से आ रहा होगा.
जी जी जी
तो
प्लीज कंटिन्यू.
अभी तक ठीक था ना? कुछ
हाँ,
प्रॉब्लम?
बहुत अच्छा है, बहुत अच्छा.
Okay, so what happens that in case of in vitro reconstitution assay, what we see that microtubules are polymerizing and go back to its seed position. So we see catastrophe. When a microtubule is polymerizing, it is called as growth phase. When it is depolymerizing, it is called as shortening phase. A transition from a growing to a shortening phase is called as catastrophe. So in vitro, we do see catastrophe. However, when a microtubule start growing from a depolymerizing end, so यहाँ पे क्या हो रहा है that microtubule go back to its original position. However, if there is something happens and microtubule start growing from here, instead of going to the original position, it is called as rescue. So in vitro, we do see catastrophe, but we don't see rescue. The I will discuss this later because there is no map here, okay, which can support microtubule to polymerize even depolymerizing effort. So MAPS or a microtubule polymerizing agent can create rescue, whereas in the absence of that, microtubule mostly go back to its original position. So with this, I started working with. So this assay is also very important, crucial to do single molecule analysis. For example, here I am showing one of the, this kinesin, which is, tracking the growing end of the microtubule and holding it. So it is causing pause event. You can see that you see that it is growing and now catch the microtubule end, and it does not allow. It means it pauses the microtubule. So you can study how many molecules of drug or molecule of a protein is present there, and do all these analysis in this assay. So this assay is very important for even doing single molecule analysis. So with this, I started with Taxol. It is one of the most successful cancer drug. Okay, even tubulin was discovered as a Taxol binding protein. Before tubulin was discovered, people were using Taxol for cancer treatment. And when even tubulin was initially purified from cell using Taxol. So this is, if somebody is interested to study the tubulin discovery and everything, they should read these old books where it was nicely showing. So it is such a old drug, okay, and it has its taxane is the dominated drug for cancer chemotherapy, used for almost all kind of cancer treatment, and it has been known that it binds to the beta subunit inside the lumen. So there are so many studies has been done. So now you will say why we are interested to do this study. So when I started in this assay and started doing it using the concentration of Taxol, which is used in cell as well as in clinic. So this concentration was found to be clinically relevant concentration, because in clinic, doctors use and cellular concentration was found to be this much thing. We started getting very surprising result. The surprising result is that, as I already discussed, that when microtubule is polymerizing, when depolymerizing, it should go back to its seed position. However, when we added Taxol 100 nanomolar, instead of going to the original position, it started growing and shrinking to a position which is far from the seed. And this seed was found to be a fixed position in the microtubule lattice. So it means that something happens here. Because of that, now microtubules are not going back to this. But there was another surprise. So surprise was this, that okay, Taxol is a stabilizer. So if it is a stabilizer, it should stabilize microtubule. If we are using very high concentration, then it should be like this, that microtubule is not able to depolymerize. It is growing, 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 growing. Or if it is a polymerizer or a stabilizer, when a microtubule is depolymerizing, it captures the end and then growing, then captures the end when it is depolymerizing, growing. Why it happens that it is only doing at one particular place? Now, why it cannot binds to here and stabilize it when this microtubule is depolymerizing? This microtubule is to go back at this position and do this. So to understand that, we know that okay, Taxol is somehow bound in the microtubule structure here. But what is really happening, to understand that, we have synthesized a green fluorescent Taxol derivative of Taxol, which is called as F-Kitex3, and started doing the turf microscope. The finding was really very surprising. You will see that Taxol, I will play this video slowly. Okay, you will see that okay, microtubule is growing, growing, growing. Now it was suddenly started growing from a position, and you will see that drug is started accumulating, and suddenly at one particular place so much of drug. So the microtubule is single from here to here, but it is not binding throughout. It is only binding to one particular place, and this binding is so much that it is after a certain time of point when microtubule will depolymerize. Suppose it was grown here. Okay, now it was depolymerized, again growing from here. It is not going back to its this position. It is going and shortening now from this position. Now I will play this video. You can see it, that okay, drug accumulated, become at one particular place. You will also see that in the beginning, so you see that there is so much of fluorescence. However, after certain time of point, the drug fluorescence decreases. So now somebody who is an expert of fluorescence or something, they will claim that it can be because of bleaching, which is called as photo bleaching. Okay, in the science, if you illuminate some sample for a long time, fluorescence can be affected because of the exposure of the light, which is called as photo bleaching. But my experiment has shown that it's not because of photo bleaching, but it is because of the relaxation of the lattice. So you need to understand that the binding is cooperative. Okay, binding of one Taxol molecule is actually supporting the binding of more and more molecule of Taxol. However, after certain time of one, you consider as a building, when a building has to be formed, a lot of labor is there. Once the building is formed, you require certain person who can just take care of the building. Is happening the same way here. Taxol is trying to modify the lattice of the microtubule, pre-existing lattice. Once this lattice is modified, rest of the drug is taken out from the lattice, and only few molecules are remain there. And by doing intensity analysis, we found that 15 to 16 drug molecules are present at the place of stable rescue sites. Okay, when we saw the EM structures, what we saw that there is a lot of defects in the microtubule structure. This was also very surprising, because Taxol being a stabilizer, it should microtubule should be more homogeneous, but we do see a lot of lattice defect in case of Taxol treatment. So we started doing FRAP assay. So what is FRAP? Is fluorescence recovery after photo bleaching. So what happens? You illuminate a section of fluorescence, and you see what happens. So you illuminate by using laser or something, and if the and if the recovery of the fluorescence is happening, so it is called as fluorescence recovery after photo bleaching, and it also indicate that the system is dynamic, interchange is happening. So what happens in case of control? For example, here there is a fluorescence, I made bleaching. Okay, here I put the laser. Now fluorescence is gone. I don't see any fluorescence recovery after that. So system is very static. Nothing is happening. So now I went to a place where drug was bound. Okay, I took a microtubule. Now identified a locus where drug was bound. You can see that drug was here, and after a certain time of point, it is relaxing. And at that place, I made a frap. Okay, you can see that the fluorescence is only recovering at the place of drug binding. You can correlate. Here is the drug binding, and fluorescence is recovering only at that particular place. So you can correlate to this part that this is the place where Taxol is binding. Although Taxol is a stabilizer, but once it is binding to that microtubule, some of the part remain open. And at that place, free tubulin can come. This was also one of the very interesting finding because till now in the tubulin or in the microtubule cytoskeleton field, it has been shown that only tubulin dimers can come from plus end on the minus end. But this data also shows that from the lattice, also dimers can come and join it. And when microtubules so are, if you next experiment which I did was ablation experiment. Okay, ablation. So frap and ablation both are laser dependent. In frap, you just hit the fluorescence, but the structure should not be deformed. But in case of ablation, you can make partial damage or complete damage. You have a very large focus, very sharp focused laser, and if the intensity is too high, it can completely damage or make the dissociation of two parts, or it can damage the part. Okay, so you can see in case of control, when I am damaging the lattice, the part is completely gone. However, when I damage the site at the place of drug binding, you will see that both parts are surviving and they can make the microtubule. And when you will see some of the examples, when accumulation of drug is so much, you will see that it will cut the part, and now it is growing it again. Now new microtubule is going because Taxol is a stabilizer. Okay, so this was something peculiar, like, okay, if defect is the place where drug is binding, we created defect in the microtubular structure. So this was one of the, Vimblastin is a depolymerizer. We used certain concentration of Vimblastin, and what we see, a lot of drug is binding. So if you compare to this structure, okay, I will just show, sorry, briefly. Okay, so only at one place drug is binding. Okay, but if you will see this chymograph, every place where defect is happening, drug is binding. Okay, so this was very, one of, I can say, one of the very interesting finding. If those people who know about the combination therapy, okay, like in combination therapy we use drugs which have similar mode of action. For example, someone is promoting cell death, another is inhibiting cell death, you will not combine. So here what is happening, Taxol is polymerizing microtubule, Vimblastin is depolymerizing microtubule. But if we know the mechanism, we can combine these two drugs, and that's what we have shown here, that two drugs having different mechanism of action on the same uh, subunit or same target, they are working together and inhibiting the tumor growth. So two drugs with different mechanism of action can also be combined together to create synergy effect in combination therapy if we really know in their mechanism of action. And to further understand what we have seen, that at the place of drug binding, where the defects are correlated with the drug binding site, we see there is a change in the protofilament number. Okay, so this is one of the very complex biophysical analysis. I am not going into the detail, but I will just give you a gist of that, that microtubules polymerizing from GMP CPP are having 14 protofilament, and in test tube, when you add Taxol, it makes microtubule 13 protofilaments. Okay, so there is a change in the protofilament number. So one microtubule is having this much of length, and when Taxol comes, so the microtubule lattice structure become less. So that is the reason, to accommodate these things, there are lattice defect into this. For example, in cells, when microtubules are polymerizing from MTOC, you all might be knowing, if you have read these cytoskeleton dynamics, that microtubules in cells are 13 protofilament, those polymerizing from MTOC. But if you will add Taxol in cells, microtubules, most of the microtubules are 12 protofilament. Because if you see this example, okay, so this is GMP CPP microtubule. Whatever microtubule is polymerizing will have 14 protofilament. Okay, 14, 14, 14. Now the Taxol will come. Now Taxol will try to make microtubule 13. Okay. Now this is 14. Now Taxol came. Taxol will try to make microtubule 14. Okay. Now two things happen here. You will, if you feel 4. So this microtubule which is polymerizing from Taxol will have 13 protofilament. Very complex biophysics and biochemistry here. But if somebody is interested, they can read these papers. Okay. Uh, and want to discuss further, I will be very happy to discuss it. But for the sake of time, I'm just giving a list. So these are the findings from my postdoc here at GBU. What I'm trying to establish my lab, or my lab is working to understand the role of autophagy. Is there something somebody would like to ask here?